Hello, my name is Tim Weil, and I'm a fellow of Pembroke College and a group leader in the Zoology department. And my lab works on Drosophila melanogaster, these what's often termed the common fruit fly. And we use this system to test different aspects of biology. And we use Drosophila for a number of different reasons. One, because they're very amenable to imaging. They're one of the premier model organisms when you're talking about working with genetics, and that we can really manipulate their genome in a way that is almost unparalleled for a system that's this advanced. And what my lab is, and myself, has always really been curious about and most interested in, is understanding how we, as trillions of cells, organized in a remarkably complex way. We have the ability to breathe. Our heart will beat our entire life, right? Our brain can remember things from decades ago, and we can bring up these memories. All of these incredibly complex functions. And we started off as one single cell. So a typical laboratory is going to have a number of different areas in them. One example would be a wet lab area where molecular biology, typical with pipetting and mixing of solutions, will take place. And in this lab, obviously, we have a nice wet lab space, as well as a sample preparation area. And so in this case, when we work in Drosophila, we're going to have a number of dissecting scopes out, and they're all going to be set up for us to prepare the tissue for the different experiments that we want to do. Now, in the case of Drosophila, we'll often have many different vials of flies with different labels on them, as you can see here, what we would term stocks. And these different animals are going to have different genes being expressed or different fluorescent proteins within their genome or different mutations. And we'll use crosses, so males and females, we'll cross them together, and this is how we can manipulate and examine the importance of different genes for different function. And when we want to work with the flies, we're going to come over to a dissecting station, and we turn on some carbon dioxide and a light source. So starting with the flies, we can then, first off, we'll take our CO2 and we'll go ahead and thread that into the bottle of animals. And as the CO2 fills up the bottle, the flies will become anesthetized. And we can see here they'll fall down to the edge. From there, we'll put them onto this carbon dioxide with porous pad. And you can see the flies will then remain anesthetized as long as the carbon dioxide is on. And what we can do then is sort males and females, for example, or pick out different phenotypes, basic phenotypes like red eyes or short bristles or curly wings. And by picking out the animals and understanding what those animals are expressing, we can then run different types of experiments. Now, a second type of experiment that we do would involve dissecting out a female and taking the ovaries from that animal. So in this case, we'd take a cover slip, we'd put it down on our dissecting scope, we'd then just put a small dab of oil down. We'll then use a couple of forceps to grab and dissect the female. And you can see here, you can simply grab these flies, then under the scope we would be able to pull off her posterior, and then we will simply remove the ovary, place the ovary into the oil, and then be able to separate out those ovaries into individual eggs, and then do different types of experimentation on those eggs. From our preparation, we would then go on to look at the sample and ask different questions. And there's two questions that we're really interested in right now in the lab. The first revolves around RNA translation. So when an RNA transcript is being translated to make protein. And we're looking at very specific RNAs and using a system that enables us to watch in real time and in a live developing tissue the first round of translation. And this is going to help us to address questions about when these proteins get made, these critical signals for patterning the egg. And then another vein that we're interested in looking at revolves around egg activation. And egg activation is an essential step in development that prepares the egg for the formation of the pronucleus. And with this egg activation is a wave of calcium, or a change in the intracellular levels of calcium, the concentration. And we're looking at examining this wave in Drosophila, trying to understand what it's used for, what's it required for within the developing cell, and also how it initiates, propagates, and because calcium is toxic to the cell, how this then recovers within the developing animal.